In the last class, we have studied about the processes like diffusion and facilitated diffusion under the chapter Transport in Plants. Today, I am going to explain you another process in passive transport, that is osmosis. Osmosis is defined as the process, or it is defined as the movement of solvent particles from a less concentrated solution into a more concentrated one across a semi-permeable membrane till it reaches the equilibrium. In the last class, we have discussed about the diffusion, which shows the movement of the solute particles. But here is the process osmosis, in which we are going to study about the movement of solvent particles. So let us recall the terms solute, solvent and solution. Solute is a substance which gets dissolved. Solvent is any liquid which dissolves the solute, and the solute plus solvent is called as solution. Let us take an example for better understanding. Take sugar crystals as solute particles and water as a solvent. Now, when we add sugar crystals to the water, it dissolves, or the sugar crystals dissolves in the solvent and forms a sugar solution. So, by this we can say that the solute and solvent when combined forms a solution. Now, coming back to the process osmosis, it is a process in which the water molecules from the dilute solution moves towards the concentrated solution. Dilute solution is not more concentrated when compared to this one. So, the water molecules or the solvent particles are moving from dilute solution to concentrated solution across a semi-permeable membrane, and this process is called as osmosis. Dilute solution is also referred to as hypotonic solution, and concentrated solution is also termed as hypertonic solution. So, the osmosis can also be defined as the movement of the water molecules from hypotonic solution to the hypertonic solution across a semi-permeable membrane. Let us see some situations in which we are going to place an animal cell and the plant cell in three different solutions. This is an isotonic solution. The solution which shows the equal concentration with that of the inner cell content is termed as isotonic solution. So, when we place the animal cell in isotonic solution, we can observe that the concentration of the isotonic solution is same as that of the concentration of inner cellular content. So, as the concentration of both these things are same, the particles or the water molecules from the solution moves into the cell, and simultaneously the water molecules from the cell or the water molecules which are present in the inner cellular content is moving out of the cell. So, in this way we can see that when we place an animal cell in an isotonic solution, the net movement of the water molecules takes place in and out of the cell. Now, in the situation 2, when we place the animal cell in hypotonic solution is nothing but less concentrated solution. Now, we can observe, or we can see that the concentration of the cellular content is more when compared to the concentration of hypotonic solution. So, we can term it as hyper. Now, we know that water always moves from hypotonic solution to hypertonic solution. Therefore, the water molecules which are present in the hypotonic solution is going to enter into the cell. When the water moves into the cell swells up and it becomes turgid because of the extra amount of the water gone into the cell. As the cell becomes turgid, and as it is an animal cell, the animal cell do not possess any cell wall or the protective layer across the cell membrane. So, due to the absence of cell wall across the cell membrane, and as the cell becomes turgid, it swells up, expand and get ruptured. Now, in the third situation, where we are going to take an animal cell in the hypertonic solution. So, when we place an animal cell in the hypertonic solution is more concentrated one. So the concentration of the cellular content is less when compared to the concentration of hypertonic solution. Now we can say that the cellular content is hypotonic. So we know that water moves from hypotonic to hypertonic. Therefore, the water molecules are coming out of the cell into the hypertonic solution. Due to this, when water coming out of the cell, it becomes shrinkage. The cell gets shrinkage and the cell becomes flaccid, or we can say crenated. In the situation 2, we have discussed that the water is going into the cell, and this condition is referred to as endoosmosis, whereas when the water is coming out of the cell, this condition is referred, or it is termed as exoosmosis. So in the situation 2, the cell is getting ruptured, and when we place an animal cell in hypertonic solution, it is going to be flaccid or crenated, and the shape of the cell gets changed due to the absence of cell wall. This is about an animal cell. When we replace this animal cell by the plant cell, then we can observe that in isotonic solution, the concentration of isotonic solution and the cellular content is same. So the net movement or the equal movement of the particles are going to take place in this situation. Whereas in the second one, where we are going to place the plant cell in hypotonic solution, it is going to show the same thing, that is, the water is moving into the cell and the condition is known as endoosmosis. But here, as in the plant cells, we can see the cell wall or the protective layer across the cell membrane. So due to the presence of this cell wall, as the cell becomes turgid, it is not going to be ruptured. It just swells up due to the accumulation or due to the extra amount of the water gone into the cell. But it do not rupture because of the presence of cell wall. Now when the plant cell is placed in the hypertonic, that is more concentrated solution, the concentration of the inner cellular content is less when compared to this solution. So now what happens, the water which is present in the big vacuole of the plant cell is coming out, and due to this the vacuole becomes smaller in size, and simultaneously the cytoplasmic content is going to come at one place. The cytoplasmic content gets detached and come at one side. Due to the loss of water present in the vacuole shrinks and becomes smaller in size, and the cytoplasmic content is going to detach and come at one side. Now this cell is called as plasmolyzed cell, and the condition where we can see the shrinkage of the cytoplasm is referred to as plasmolysis. Now when we take out this cell and again we place it in hypotonic solution, then the cell regain all its water again, and this process is called as deplasmolysis. Deplasmolysis is a process where we can place the plasmolyzed cell into the hypotonic solution so that the water moves into the cell and the shrinkage gets recovered, or the cell will regain its water. So this process is called as deplasmolysis. In case of animal cell, in one situation the cell is getting ruptured, and in the other situation the shape of the cell gets changed due to the absence of cell wall. But in case of plant cell, during the endoosmosis, the cell is not going to be ruptured because of the presence of cell wall, and also in the condition of exoosmosis, the cell do not change its shape because of the presence of protective layer across its membrane. Let us move to the next process, that is imbibition. Imbibition is a process in which adsorption of water takes place. Now what is the meaning of adsorption? Adsorption, we are not talking about absorption. Here the adsorption of water takes place in the process of imbibition. So adsorption is the process in which the solid surface holds the liquid or the gaseous molecules. So this is called the adsorption of water. So imbibition is a process in which the water is getting adsorbed on the surface, and when we talk about the process imbibition, some common examples comes in our mind, and that are, if we soak the seeds into the water, the seeds imbibes the water and swells up. Another example is during rainy season, the wooden doors get swollen, or the wooden doors swells up due to the imbibition. So these are certain examples that we can see due to the process of imbibition. The imbibition shows certain changes, and that are, the first one is, imbibition is useful in generating pressure. It helps in producing some pressure, and that pressure is known as imbibition pressure. This imbibition pressure is very useful because when a dry seed is fall in a thin crevice of the rock, during rain, this dry seed imbibes water and it swells up, and when it swells up, it generates some pressure. The dry seed can generate up to 1000 atmosphere of pressure, which is enough to widen this crack, and it helps in the soil formation. The next change that we can see in the process of imbibition is heat is generated or heat is released, and this heat which is generated in the process or by the process of imbibition is called as heat of hydration, or it is also called as heat of wetting. As the water molecules are involved in this process, that's the reason it is called as heat of hydration or heat of wetting. Suppose this is the surface on which the water has to imbibe, and these are some water molecules which are moving randomly in different directions, and these moving water particles have some kinetic energy. When these moving water particles is getting absorbed on this solid surface, this kinetic energy is getting converted into heat energy. So in this way, the heat is released in the process of imbibition. The next change is, the imbibition helps in increasing the volume, or it helps in changing the volume. So in the beginning, we have studied one example that during rainy season, the wooden doors swells up due to the imbibition. So in this way, it shows that the volume of the doors are getting changed, and we find difficulty to close it. This difficulty or the swelling of the doors is due to the imbibition. As the doors are imbibing water during rainy season, it swells up and it changes its volume. So these are some of the changes which we can see during the process of imbibition, and they are imbibition pressure, heat is released, and the volume is changed. The next topic is water potential. It is an important term which can be used when we study the transport in plants. So the water potential is the potential of water molecules to do work, and it is represented by the symbol psi, and the units which are used to measure the water potential is pascals. Now when we observe the water molecules in a medium, it moves randomly in different directions. As I said you before, that these moving water particles or the water molecules have kinetic energy, and when we take it as a pure water, then we can see that maximum number of the water molecules are present in the pure water. When there is a maximum number of the water molecules are present in the solution or in the pure water, then definitely the energy which is present or which is possessed by the water molecules is also maximum. So the water molecules have maximum energy. If the energy is maximum, then ultimately the potential which is used to carry out the work is also maximum. So in the pure water, as the number of the water molecules are more, the energy is maximum, and the potential which is present in the water molecules is also maximum to carry out the work. The water potential is represented by psi W, and the water potential of pure water is maximum, and it is taken as 0. So the maximum water potential of the pure water is taken as 0. Now when we add some solutes in this pure water, then it becomes solution, and in the solution we can see that there are no more amount of the water molecules are present. Hence the energy and the potential which is possessed by the water molecules are less when compared to the pure water. So the water potential of solutions is always less than 0. As it contains solute in it, it becomes the water molecules become lesser in number, and hence the energy is also becomes minimum, and it is always less than 0. We can take -1, -2 and so on. So this is about the water potential of solution and water potential of pure water. Let us talk about some factors that affects the water potential. There are 3 factors that can affect the water potential, and they are solute potential or solute concentration, we can say. The next is pressure potential, and the third one is gravity. As we know that the water potential of the pure water is maximum and it is taken as 0. But when we add some solutes or some solute particles in the pure water, then it becomes solution, and due to the concentration of the solute, the water potential gets decreases. Therefore, the concentration of the solute, if the concentration of the solute increases, the water potential decreases. So it shows the negative effect. Coming to the next one, that is pressure potential. The pressure potential is represented by psi p, and solute potential is represented by psi s. Now, when the pressure potential is increased, the water potential is also gets increased. So this shows the positive effect of pressure potential on water potential. The last one is gravity. This factor is negligible, so we can avoid it because it does not have any effect on the water potential substantially. So we can avoid it. By these factors, we can calculate or we can show a relation that helps to calculate the water potential of the liquids. So the water potential can be calculated by this relation, that is psi w is equals to psi s plus psi p. This is an equation which is used to calculate the water potential. And one thing we need to remember, that the water always moves from a region of high water potential to low water potential. When we talk about the water transport from the soil towards the leaf, then the entire process is going to take place by 2 steps. First step is absorption of water, and the second one is ascent of sap. In the first process, that is absorption of water, we can study about how the water is transported or how the water is absorbed from the soil by the roots. Whereas in the second step, that is in ascent of sap, we can learn about how the water from the roots is getting transported in the upward direction to the leaves. Firstly, we will talk about or we will discuss about the first step, that is absorption of water. So to study about the absorption of water, we need to observe the clear structure of root. So the clear structure of root or the closed structure of roots shows that the epidermal cell of the roots have some protrusions, and these protrusions are called as root hairs. The fluid which is present in the root hairs is called as cell sap. After the epidermal layer, some cortical cells are present, and these cortical cells are loosely arranged. As they are loosely arranged, we can clearly see some spaces in between the cells, and these spaces are called as intercellular spaces. Next to the cortex, one layer is present which is termed as endodermis, and this layer of cells shows some thickening in the walls of the cells, and these thickenings is composed of material called suberin. Suberin is a material which is impermeable to water. So, and the layer of this thickening walls is termed as Casparian strips. In the endodermis, some cells are present without these thickenings, means without this material suberin, and the cells which are without these thickenings are termed as passage cells. These passage cells acts as a barrier for the movement of water towards the inner side. The next to this endodermis, the layer of cells or the compact cells are present, and this forms pericycle. Inner to the pericycle, sieve tube cells are present, which is called as phloem. And inner to this phloem, if we talk about the dicot, then we can see a layer of cells or the network of cell which is called as cambium. Cambium is a layer which is present in between the two types of conductive tissues, that is in between the phloem and xylem. So, after the cambium, the cells which are present are called as xylem. The water which moves from the soil, it has to reaches to the xylem vessels. So, now how the water which is absorbed from the soil is going to reaches the xylem? The movement of the water takes place by two means. They are apoplast and symplast. The apoplast movement shows that the water moves only through the cell wall or through the intercellular spaces. The water cannot move inside this membrane. It cannot cross the membrane of the cells. Only moves through the cell wall or through the intercellular spaces. So this type of movement is known as apoplast movement. When apoplastic movement or apoplastic pathway is taking place, if the water is getting inside the cell, it won't happen. But if the water is getting inside the cell, then that water is combining with the cytoplasm and it forms a solution. So that will show the symplastic pathway. So the apoplast movement can also be defined as the movement of the water molecules without forming a solution. The water which is present in the soil first absorbed by the root hairs, it moves from the root hairs and reaches to the intercellular spaces of the cortical cell, and then from these intercellular spaces it reaches to the layer of endodermis. When it reaches to the layer of endodermis, if it reaches to the thickening, that is near to the Casparian strips, then these are the barriers which do not allow the water to enter it. But if it reaches the passage cells, the passage cells are without thickenings. The suberin material is not present in the passage cell, as the suberin material is impermeable to water. It does not allow the water to move in. So when the water reaches these passage cells, it can transport it to the other cells of the roots. So this is about the apoplastic movement of the water, and the apoplastic pathway shows or it helps in the maximum amount of the water transportation. The next one is symplast. When we talk about the symplast, it shows that the water is going to move into the cell. It can cross the membrane, and from the root hairs, when the water from the soil enters into the root hairs through osmosis, this water is going inside the cells. It crosses the membrane, it goes inside the cell, then reaches the passage cells, and from these they can easily enter into the other cells of the roots. But when we talk about the apoplastic movement of the water, when the water reaches through apoplast towards the passage cells, these apoplasts crosses the membrane of the passage cells, and then after getting out of these passage cells, it again show the apoplastic movement till it reaches the xylem. So the water which is moved through two means, that is through apoplast and symplast, it has to reach to the xylem vessels. When the water reaches to the xylem vessels through apoplastic pathway and through the symplastic pathway, it is going to transport it in the upward direction to the leaves. So these xylem vessels are helping in the upward movement of the water in the plant. The things we need to remember is that the maximum absorption of the water takes place through apoplast, and the less amount of the water is getting absorbed through symplast. And also that the cortical cells shows both apoplastic movements and the symplastic movements. The water which is moved from one cell to another cell, it is going to take place through some specialized pores which are present in the cell wall of the cortical cell, and these pores are termed as plasmodesmata. So the water moves from one cell to another cell through plasmodesmata, and in the cortical cell both apoplastic and symplastic movements can be seen, whereas in the layer endodermis only symplastic movements can be seen, and on the inner again both the apoplastic and the symplastic movements occur to absorb the water. We know that the water moves from a region of high water potential to lower water potential. So let's see that how the water is going to be absorbed from the soil by the roots. The soil contains water and this water is hypotonic, that is, it is less concentrated when compared to the cell sap present in the root heads. So the cell sap is hypertonic and the water which is present in the soil is hypotonic, and the water moves from hypotonic to hypertonic. So in this way the cell sap absorbs the water present in the soil. Then from the cell sap, the water which is absorbed by the cell sap is less concentrated when compared to the cells present in the cortex. So these cells shows higher concentration. In this way, the difference in the concentration helps the cells to absorb the water from one cell to another cell, and the water moves from one cell to another cell through plasmodesmata, and also because of the difference in their concentration. These cortical cells absorbs the iron compounds or some other materials from the surrounding, and these materials helps them to increase its concentration. So when we compare the concentration of cell sap with this cell, we can see that the cell sap becomes hypotonic when compared to the cortical cell. In this way, the water moves from hypotonic solution to hypertonic solution, and the absorption of water takes place from one cell to another cell, and then to the next, and then to another cell. So in this way the water moves till it reaches the xylem vessels, and from the xylem vessels, as they are the conductive tissues, the xylem vessels helps in the upward movement of the water. It transports the water to the leaves. So this is about the first step in the movement of water, that is about the absorption of water we have learned. And in this step we have studied about two means, that are apoplast and symplast. Apoplast is the mechanism, or it is a means by which the water moves through the cell wall or through the intercellular spaces. Symplastic movement shows the water movement into the cells and then it reaches to the xylem vessels, and from the xylem vessels it shows the upward movement of the water. This is about the first step, that is absorption of water. The second step, that is ascent of sap, how the water is going to move upwards from the xylem vessels, we'll discuss in our next